Hi, AP Bio students. Welcome to Chapter 5, all about the cell membrane, or otherwise known as the plasma membrane. In this chapter, we're going to be talking about what does a membrane look like, what is its structure, and its function. So let's begin with a little picture. Let me fix my screen here. All right, so the first thing we want to look at, and this picture's in your textbook, is artistic depiction of the plasma membrane. Now, this picture shows two cells in your brain, two brain cells. And the question that we want to look at today is, how do your brain cells communicate with each other when you're learning? How does one cell send a message to another cell? What's going on when that happens? Well, the details of it we're going to save for later, but for now, something important to note here is that in order for cells to communicate with each other, they need to send molecules between one cell to another cell. So if you look here we have little vesicles, and these vesicles have molecules inside, and these vesicles are going to end up reaching the membrane of the cell, the outside of the cell, and the vesicle will fuse with the membrane and it will secrete the molecules inside into the space in between the two cells. And these molecules are going to bind to these light yellow, light green molecules here, which are proteins. These are called receptor proteins. And these proteins are going to receive the molecules, and there will be a signal that will be sent to this cell. Basically, this cell knows that something's happening over here, and cells are sending messages with each other through different types of molecules. And we'll talk more about this in this chapter, but that's how your cells can communicate with each other. So we're talking about the plasma membrane. And why are plasma membranes important? Well, cells are the smallest, most basic unit of life, and there's a very clear inside and there is a very clear outside. So the plasma membrane, or the cell membrane, is like the boundary that separates a cell from its environment. Now, can the cell membrane be impenetrable? What does that mean? That means nothing can get in and out. Should it be impenetrable? Don't you think that cells should protect themselves? Shouldn't they keep everything inside? The answer is no. Why should cell membranes not be impenetrable? That's because stuff needs to get in and out. That's how cells work. Cells need materials in, and then products, the stuff that they produce, like proteins or messenger molecules or waste, need to get out. So, some things that need to get in are food, so the cell can have energy, carbohydrates, sugars, proteins, amino acids, lipids, salts, oxygen, water. When we talk about metabolism, we talk about cellular respiration. We'll learn about why all these components are important. But basically, all these materials help the cell stay alive and give it energy, supply it with nutrients. And then stuff needs to come out. Waste, ammonia, these are waste products. We'll learn more about when we get to cellular respiration. Salts, carbon dioxide, water, and some other products that are going to be shipped out and sent to other cells or to help the cells communicate with each other. And then remember, that's why it's so important for cells to have a high surface area to volume ratio. So the higher the surface area compared to the volume means stuff can get in and out at a more efficient rate. Okay, so how are we going to build our membrane? If you had the option to build a barrier that keeps all the watery contents of the cell inside, separate from the watery environment outside. So inside the cell, we've got some water inside, and even outside the cell, cells are often um, bound in water. And so, what kind of molecule should we build the membrane out of? Should we build it out of carbohydrates? Think about the structure of a carbohydrate. Should we build it out of proteins? Think about how these macromolecules will interact in water. Carbohydrates, lots of carbons, hydrogens, oxygens. Proteins have the amino end and the carboxyl end, and they are able to, they have like charged ends. So one side is like more positively charged, one side more negatively charged. Think about how that will interact with water. Think about carbohydrates, that oxygen, that very electronegative atom. How will those atoms interact with water? If you said that they'll probably dissolve, you're correct, because water is a polar molecule. Remember, proteins and carbohydrates have polar sides. They have, some sides are more charged than others, and so the water can easily attach on and bind on to these two molecules and break them apart. So that wouldn't really work if you put a carbohydrate or a protein just straight in water. Because water's polar, it will bind to these macromolecules and kind of dissolve it. Nucleic acids, same. Nucleic acids have polarity. There's a lot of charges within the macromolecule, so those will be destroyed by water. But lipids, what do you know about lipids? Remember, lipids often have a hydrophobic side to them because lipids are really just a long chain of carbon and hydrogen. Carbon and hydrogen have pretty much equal electronegativity, meaning there's not like one more positive or one more negative side of the molecule, which means they're hydrophobic. They don't really attach to water. Because water is polar, water likes to attach to molecules that have some sort of charge. But because lipids are hydrophobic, because lipids are nonpolar, they're mostly just straight up hydrogen carbon, they don't really act like magnets. So the water and the lipids don't really mix together. So that's why we use lipids to build our membrane around the cell, because oil and water don't mix. So it really does keep that layer around the water intact and doesn't dissolve. The lipids aren't dissolved by the water. So the membranes are made up of lipids, and specifically phospholipids. So phospholipids, we have a glycerol head. And remember, the glycerol head, we'll look on the next slide, is polar, and it has a slightly negative charge, but it has two fatty acid chains for the tails. So here's the fatty acid tails, here's the phosphate head. It has a phosphorus, which is slightly electronegative, or pretty electronegative as an atom. And so this end will end up liking water because it has a slightly charged part of it. So it will want to bind like magnets with water. So it's hydrophilic. Philic means love. Whereas the tail, it's just a bunch of hydrogens and carbons. This side will be hydrophobic. It's nonpolar or hydrophobic. And so if you placed one of these phospholipids in water, well, the hydrophilic side, the side that likes water, will be facing the water. So inside and outside the cell, let's say there's water. Well, you are going to have the hydrophilic side, the head facing the water. And we know that the membrane is a bilayer. Bi means two. So we have one row of phospholipids on top and another row on bottom. And notice on the bottom, here's water outside the cell. And here's the hydrophilic heads, the water-loving heads facing the water on the outside. And then the tails are protected from the water because they're hydrophobic, they're nonpolar, facing inwards. So now you have this bilayer, two layers of phospholipids. So let's look really close again at the structure of a phospholipid, because this is where we're kind of combining multiple units and it might be a little bit complex. So let's look at the head right here. So the head is hydrophilic. Hydrophilic means water loving, and it's hydrophilic because notice here, if you look really closely, this is really important, you see that negatively charged oxygen. So this negatively charged oxygen, imagine if you were to take a water molecule, the positive side of the water molecule, the side with the hydrogens, if it came near this hydrophobic head, if it came near this oxygen, this negatively charged oxygen, they're going to come together like magnets. And so this head, this part of the molecule likes water, it will bind with water. Whereas the hydrophobic tails are just made of a bunch of carbons in black and hydrogens in gray. And so these tails are going to not act like magnets with water because they're just pretty much neutral charge all over. All right, so we kind of talked about this before, but just to kind of see it up close, here's a bunch of water molecules. And notice we have oxygen and hydrogen. Remember, water is polar, so it has a slightly positive and a slightly negative end. And notice right here, this is the phosphorus atom that has the oxygen attached to it. And so here, this oxygen right here, that's negatively charged, is going to be attracted to these hydrogens in gray. And so they're going to come together like magnets. And if you see here, actually even here we have a nitrogen that's a little bit that's positively charged. So if you look up here, the negative red oxygen on the water is going to be attracted to the positively charged nitrogen up here in the head up here. But down here we have a bunch of just carbons and hydrogens, and so they're going to face away from the water. They don't want anything to do with water because water is not going to stick to the hydrocarbon chain. And so what you end up getting is this beautiful little bilayer. Okay, now the membrane is a little bit more complex than just a barrier. And scientists are still learning more about the membrane of cells. They learn more every year. And so it's not just these phospholipids. It actually includes a bunch of other things. So let's take a little tour of the membrane. And I remember the first time I learned this, I thought it was really cool just to see up close. Just the complexity of this, I think, is really beautiful. So just take a moment and look at this picture. Notice you can see the phospholipid bilayer. So you see the hydrophilic heads. Remember, philic means loving. So water-loving heads, hydrophobic tails, and our bilayer. And then we have our intracellular space. So intra means inside the cell. And then here we have our extracellular space, so outside the cell. And here you can see even outside the cell, there's stuff going on. There's these fibers that are kind of moving around that the cell can attach to. So you see here, it's the cells attached. And so everything's kind of held together. And what we're looking at now is actually an animal cell. So notice here that there's no cell wall outside like prokaryotic cells have and plant cells have. So we're just looking at animal cells here. So this could be one of your cells. And let's take a little tour. So starting in the top left, again, we see the fibers here. I told you of the extracellular matrix. Extra means outside the cell, and matrix is basically just like the space. And notice here that this molecule right here, which is a protein, it is connected to the fibers. You'll see inside the cell we have microfilaments of the cytoskeleton. So this is like holding all the organelles in place inside the cell. We have our phospholipids here, and embedded within the phospholipid bilayer, we sometimes have these special molecules called cholesterol. And cholesterol is a special type of lipid that are embedded in the membrane. Here we have a protein that is attached to a carbohydrate, so some carbohydrates attached. And the carbohydrates act as kind of like tags for the molecule or for the cell. So if there's another cell around it, this kind of acts as like, hey, this is my barcode. This is who I am. Kind of like a name tag for the cell. So other cells are like familiar with who it is. Let's see, what else do we have? Okay, we have glycolipids, the extracellular side, so it's outside. So the lipids or the phospholipids that are facing out are called extracellular side. The phospholipids facing inside are called the cytoplasmic side because they're facing inside in the cytoplasm. We have some proteins that are integrated into the plasma membrane. So this right here is called an integral protein. And then some are on the periphery. Periphery means the outside, or they're kind of like right underneath the cell membrane. And even some can be on top as well. So they're kind of hanging out on the outside. All right, so when you think of the plasma membrane, I want you to think of the membrane as like this fluid membrane that's like all moving around. So if you were to put the cell in like water, notice that it's kind of like up and down, up and down. And that's because the membrane is kind of moving around. It's very fluid. It's not rigid. It's not just like all intact. It's a fluid membrane. And there are a couple different factors that affect fluidity. So the first factor that affects how fluid or rigid the membrane will be the nature of the hydrocarbon tails of the phospholipids. So, if you remember from Unit 1, lipids can either be saturated or unsaturated. So, saturated means if you have a bunch of carbons in a row, the carbons are all attached to hydrogen atoms. And if every single carbon is attached to a hydrogen, that's called a saturated fatty acid, or the tail here. Now, if you have two carbons that are bonded with a double bond, it kind of bends the tail a little bit, and because there's a double bond, the two carbons won't be fully surrounded by hydrogens. So go back and review saturated versus unsaturated fatty acids if you need to. But basically, because there's a double bond here, it will bend the tail. This is called an unsaturated fatty acid. So that bend in the tail, notice what it does to the membrane. Do you see all this extra space in here? What that will do is it will actually make the membrane even more fluid. So the phospholipids can't pack really closely together, and so it will enhance the fluidity of the membrane. It will be more fluid. Versus if you had a bunch of saturated hydrocarbon tails, so these fatty acids, if they're just a bunch of hydrocarbon chains without double bonds, so basically just straight chains here, notice how you can pack in the membrane or the phospholipids really, really tightly, and that will keep it super, super, super or less fluid, and we call that viscous. So if it's less fluid, if it's like harder to move, we call that viscous. So it increases the viscosity or the ability to move of the membrane. And the last way that you can affect membrane fluidity is actually by increasing the amount of cholesterol. So remember, cholesterol is another type of lipid, and this is found in animal cells. And what this does, it actually changes the way that the membrane will respond to different temperatures. So at normal temperatures, at moderate temperatures, this cholesterol actually keeps the membrane a little bit more rigid or less fluid. So it kind of keeps it more intact. Like think of these as kind of like these like walls that hold everything together. So it's the tails are usually like flopping around, but because the cholesterol's in there, it's a little bit tighter and it keeps it all structured and more. But let's say you cool down your cell, or your cells is in really cold environments. Well, when you lower the temperature, it causes molecules to actually slow down their movement. And so if molecules are slowing down, then typically, like let's say you had this membrane here, then that would be really tightly packed because the tails would be not moving around as much. They'd just be really tightly packed and like frozen kind of. But for this kind of plasma membrane, if you had a really low temperature, remember the molecules are kind of slowing down a little bit, it actually stops them from completely solidifying. So if you lower the temperature too much, this is why living things can die in really cold temperatures, because your cell membranes are going to really like freeze up. They're going to stop being fluid, and that could cause problems for your cell. And so cholesterol actually keeps the membrane from packing too tight. So that's the benefit of having cholesterol. And animal cells have this cholesterol, so we can survive in colder environments. Okay, so this is actually connected to evolution. So different types of living things have different membrane compositions. What that means is which living organisms would have this type of membrane versus this type of membrane versus this type of membrane. Um, well, the idea here is that certain living things can actually respond to the temperature changes that they've evolved to live in. So, for example, if you are an organism that lives in a really cold environment, then you might want to have a more fluid membrane, right? Because you don't want your membrane to become packed too tightly. If you are living in a hotter environment, then you might want to have a more tightly packed membrane, because you're not at the danger of your membrane freezing up. It's because you're living in a hot environment, so you don't really need to protect your membrane in that way. And so scientists have found that depending on where organisms live, their membranes kind of match the environment that they live in. All right, so far in this video, we've talked about the lipids that construct the cell membrane. So we have the phospholipid bilayer with the hydrophilic heads that are facing out towards the aqueous outside of the cell, and also the aqueous inside of the cell. Those are the heads, hydrophilic. And then the hydrophobic tails, the fatty acid chains, the hydrocarbon chains that are facing inside because they're trying to escape water. They're hydrophobic. And in this video, we're going to look at You Must Know number 2 for chapter 5, which will talk more about the proteins and the carbohydrates that are found in membranes. So if we look at our picture here, let's orient ourselves. Let's take a little tour of the membrane. We've already talked about the lipids, but now in this video, we're going to focus in on these proteins. So you see in purple, these proteins are embedded, and some of them are even outside. And then we even have carbohydrates that are embedded in the membrane as well. So if you see up here in green, this carbohydrate is bound to a protein. We call that a glycoprotein. And so we'll take a look at the function of those in this video. So first, let's take a look at the structure of these proteins. If you were to zoom into one of these proteins, what would it look like? And we call this a transmembrane protein. Trans means across in Latin, the membrane. So this protein is across the membrane. And if you zoom in really close, you can see the secondary and tertiary and quaternary structure of this protein. Remember, made up of amino acids. We have the N-terminus side, the amino side sticking out on the top, and the C-terminus side sticking out on the bottom of the membrane. Now, there are different types of proteins, and they have different functions within the cell membrane. So the first type are called integral proteins. Integral proteins are integrated within the cell membrane, and they penetrate the hydrophobic interior. Remember, the interior here is hydrophobic. These fatty acid tails are escaping water. And so remember how we talked about how different amino acids, remember there's 20 of them, they can have different, they can either be hydrophobic or hydrophilic or charged or not charged or polar or nonpolar. And so all of the amino acids that make up the inside of the protein are going to be hydrophobic or nonpolar because they're able to kind of sit within this hydrophobic space in the middle. They're called transmembrane proteins, and they consist of stretches of nonpolar amino acids coiled into alpha helices. So we just talked about that. This is the nonpolar section of the amino acid, or of the protein. Then there are peripheral proteins. They're loosely bound to the surface. So these peripheral proteins are kind of on the outside. All right, now there are 6 major functions of membrane proteins, and we're going to talk through each of these very briefly. And we're going to be talking a lot more about the functions of these proteins. So I'm just going to, the purpose of this is just kind of give you an overview. It's not super important that you remember every single detail of this right now, because as we go throughout the rest of this entire year, we're going to be coming back to a lot of this. But the first, and I'm going to go through this kind of quickly, we have our transport proteins. So transport proteins allow things to come in and out of the cell. Now, sometimes things can come in and out very quickly, but sometimes things need energy. They need to be supported with energy, or ATP, which is a form of energy in the cell, in order to allow things to come in and out. So transport of molecules or ions in and out of the cell. And we'll talk more about that later in this video. Enzymatic activity. So sometimes there are these proteins called enzymes inside cells, and the proteins in the cell membrane will actually help kind of change the shape of these enzymes. So notice here, this is one enzyme that has a certain shape. It's going to bind here to the protein, and it will actually change its shape. So it'll come off as a square, and then that action will allow it to bind to the other side of the protein, which will change its shape into a circle, and then that circle can be used for other places in the cell. It's a really cool concept. We'll talk a lot more about this. This is connected to when we talk about cell signaling and enzymes. We'll talk more about that. Same here. Sometimes there are signals or signaling molecules, for example, hormones, we'll talk more about that later, that come from other places in the body, and they need to send a message to the cell. So the molecule will bind to this receptor protein, and a signal transduction will be, basically that means there's going to be a chain reaction of different things that happen within the cell that basically communicates to the cell that there's something outside that's happening, and something will happen inside the cell because of this signal. So cell-to-cell recognition. So remember we talked about those glycoproteins. These are carbohydrates that bound to proteins. This is the function of carbohydrates within the cell membrane. So this is a key part of your you must know. These glycoproteins, or these carbohydrates here in green, allow 2 adjacent cells to recognize each other. So you'll have a protein here and a protein here. And if that carbohydrate is in the center, then the cells know that their neighbor is near. They're kind of like holding hands. Intercellular joining, so sometimes 2 cells can be joined together by 2 proteins that are connected, and then even proteins can attach to the cytoskeleton. So the cytoskeleton are these fibers outside in the extracellular matrix. Extra means outside the cell, matrix means all the stuff that's surrounding it, and so these proteins attach on and kind of hold the cell in place within the space outside the cell. Okay, so we just talked about this a little bit, but we talked about these carbohydrates that are found within the cell membrane. We call them glycoproteins. And again, this is how one cell is able to kind of recognize its neighboring cell. This is important in organ and tissue development because your tissues have cells that are really, really close together and they need to communicate with each other. And so these carbohydrates will help keep these cells kind of aware of who's next to them. And this also is connected to the idea of blood types. And so there are different blood types, and this is the reason why it's important that you're getting the right blood. If you're getting a blood transfusion, that you get the right type, because on the surface of your blood cells, you have carbohydrates that are kind of like giving the red blood cell its identity. So group O, we see here, has carbohydrates, but they're very simple. Group A has the same exact structure here as group O, but it's added on another molecule here. So this is another part of the carbohydrate, extending it longer. Group B has a different shape, and group AB, notice, has both of them. So both of these, this one and this one, together on the surface. And so if you have group A blood, then you are going to have these carbohydrates on the surface. And so if you were given group B blood, type B blood, and you were given it to someone with type A, then you would have a whole new carbohydrate on the surface. And we'll talk more about this later, but there are these specialized molecules within your immune system called antigens. And if they don't recognize the right carbohydrate in your blood, then they will attack that blood and cause it to clump. So basically, just connecting to blood types, these are carbohydrates on the surface of red blood cells that allow your blood to kind of recognize neighboring blood cells and make sure there's no invaders. All right, so we've talked about the phospholipid membrane, and we've talked about proteins, we talked about lipids, and the carbohydrates that are within them. In this next part of the video, we're going to talk about cell junctions. A junction is the place where two cells are coming together, and this is actually the end of chapter 4 in your textbook. So if you already read that, great, but let's kind of review how two cells that are neighboring each other are communicating. So we're first going to talk about plant cells. Now, plant cells, remember, have a cell wall around their membrane. And let's look at the structure of the plant cell wall and then how two plant cells will communicate with each other. So first of all, plant cells, the cell wall is made up of molecules called cellulose, which is a type of carbohydrate. And there are multiple parts of the cell wall. So there's the primary cell wall. If we look here, it's the space that's closest to the inside, or the space in between two cells. So if we look at this picture here, here's cell 1, and then this kind of middle section that's between the cells, and then here's cell 2. So the primary cell wall is like really, really close. It's like on the outside. Then you have the secondary cell wall. So it's like kind of next to the primary cell wall. And there can be multiple layers. And plant cells that kind of are a little tougher or harder, like wood, has multiple layers of secondary cell wall. And then in the center, you have the middle lamella, and the lamella is basically the part between two cell walls, and that's made up of a bunch of sticky polysaccharides, so sugars, and that's in between. All right, so if you look here in this picture, here's a bunch of cells, plant cells, and their cell walls, and they've got this middle lamella between them. We're going to talk on the next page about, okay, so how does one cell communicate with the next? And we'll talk about this idea of plasmodesmata. So intercellular junctions. Intercellular means between two cells. How do they communicate? So for plant cells, they have this thing called plasmodesmata. Say that to yourself, plasmodesmata. It's kind of a fun word. Basically, plasmodesmata are channels. They're made up of membranes, so it's basically like a tunnel, basically between, or a channel between two plant cells, allowing cytosol. Remember, cytosol is a fluid inside the cell to pass between the cells. So whatever's in here, any proteins, any molecules, small particles, small molecules can pass through the channels. So here again is the plasmodesmata. So here's one cell on the left, here's one cell on the right, and the plasmodesmata is like this membranous channel that allows, basically the two of them are connected. They're like holding hands with each other and allowing things to go between them. Okay, and that's really important actually because they're communicating. They're sending nutrients to each other, they're sending signals to each other, and so all the cells are connected. It's not just every cell on its own, they're all communicating with each other. And we'll have a whole unit where we talk all about cell communication and how they communicate and what kind of molecules they send. But just for now, no, plasmodesmata is how two plant cells communicate. And again, here we see the primary cell wall kind of on the inside, the secondary cell wall on the outside. There can be multiple layers in the middle lamella with the plasmodesmata in between. Here's a picture under a microscope. Here's two cells, one on the top, one on the bottom, and then here is the lamella in between. And you can see the plasmodesmata going across, allowing them to communicate with each other. Okay, so we just talked about plant cells. Now let's talk about animal cells. So remember, we talked about the surface of the membrane is connected to something called the extracellular matrix. Extra means outside the cell. And it's a bunch of fibers made up of a protein called collagen and a network of glycoproteins, so a bunch of carbohydrates. So this right here, a polysaccharide, that's a carbohydrate connected to a protein here. This is a fiber or a protein called collagen. And you can see the proteins that are embedded can actually connect to the extracellular matrix holding it in place. So it allows the cell to be supported within the matrix. Adhesion, so adhesion basically means they're connected, they're sticking to each other. It can allow the cell to move around, so depending on how the extracellular matrix is moving, it can move around. And also regulation, so depending on what's out here can determine what is coming into the cell. Okay, now intercellular junctions in animals. Remember, animals don't have these cell walls, so it's a little bit easier for animal cells to communicate through their membranes. And there's three main types of junctions in animals. And the specific details aren't super important, but just so you can see, there's three types. The first type is tight junction and desmosomes. These are very similar. The tight junction, basically, they are these kind of openings here, and they are made up of the cytoskeleton within the cell, and they prevent fluid from moving across the layer of cells. So basically, they kind of keep the two layers between the cells sealed together. Then we have desmosomes also providing structure so that the two cells stay attached. So basically kind of keeping attached two cells together. And then this part is the more important part here. They are called gap junctions. So gap junctions are openings between the two cells that basically allow really small molecules or ions, small charged particles to pass through between two cells. Here's a desmosome. So right here, here's one cell on the left and one cell on the right, and desmosomes are holding the cells together. Here are gap junctions, again, holding the two cells together. And then finally, the gap junctions, are allowing small ions to pass through between the two cells. And then we have tight junctions again, which kind of like the desmosomes are holding two cells in place, keeping them connected. Okay, so we talked just now about how cells are communicating, but I next want to show you how larger molecules, let's say you're not an ion, how can you get out of the cell if you can't fit through the gap junctions? So we kind of talked about this when we learned about the endomembrane system, and we talked about how proteins are built in the endoplasmic reticulum and the ribosomes, and then pass through the Golgi apparatus, and then are make their way out of the cell in these little pods called vesicles. So just to recap, we call these little pods vesicles and vacuoles, and it's how you get stuff in and out. So basically, the word for this is endocytosis. Endocytosis. Endo means in or within. Cyto means cell. And so phagocytosis is one type of endocytosis. And basically that's cellular eating. So if there was something outside the cell, let's see if I have a picture. I do. So let's say there's food outside the cell and it needs to get in. Then it will kind of sit on the surface of the membrane and it will fuse, and the membrane will kind of fuse together and create this vesicle. And then eventually that vesicle will fuse with a lysosome. Remember, the lysosome can lyse up larger molecules and make them smaller. And then those are molecules that can be used for your cell later on from the mitochondria to get energy from them, make ATP. We'll get to that when we talk about cellular respiration. Pinocytosis, the fluid in the extracellular matrix, if it's coming in, these ions or really small particles or water, basically anything that's dissolved, not these larger sugar molecules or food molecules, they can get in and do whatever they need. There's not like a specific process that happens next, but those can come in. And then sometimes receptor-mediated endocytosis. So this is when certain molecules need to bind to certain carbohydrates or proteins within the surface of the membrane, and that will form a vesicle. And then this will go on, and it will have a specific function or purpose that it's getting to because it's been accepted by this receptor that knows where it needs to go next. And we'll talk a lot more about that when we get to cell signaling. So I'm going to skip this for now. We'll come back to it. All right, so we've talked about the plasma membrane. We talked about the structure of the plasma membrane. So we know it's made up of phospholipids that make up the phospholipid bilayer, proteins, integral and peripheral, and then these carbohydrates basically function as cell recognition between two cells. And now we're going to talk about how things can get across the membrane. So if you have something out here, something inside, and if it's not a large molecule doing endocytosis, then how can we get other molecules in and out? Okay, so the first thing we need to talk about is some physics. And the physics is the second law of thermodynamics, which applies to biology as well. Physics also applies to biology. That's why you learn physics, because it's all related to how biology works. So basically, the second law of thermodynamics states that the universe tends towards disorder. If I take an egg that's not cooked and I crack it into my pan, can I get that egg and put it back into its shell and put it back together? No, it's become more disordered. And that's just one example. There's many examples. When you take physics, you'll learn more this in more detail. But the idea of the universe tending towards disorder is called entropy. There's lots of more to learn about this. We're just skimming the surface. But basically, let's take a look at an example. Let's imagine you had some dye. And the dye, you can see really closely, we see each individual molecule of dye. And then we put a membrane within the dye. So let's say this is like a beaker right here, and there's a membrane, and this is a cross section, so it's going up and down. And on the other side, we just have plain water. There's no dye. Well, what's going to happen over time? Well, notice that the molecules actually are small enough to get through the spaces between the membrane because there's spaces in between, so it can get in and out. And they're kind of going back and forth. But eventually, according to the law of thermodynamics, the universe tends towards disorder. At the beginning, all the molecules were nicely, neatly packed on one side, all organized, kind of like your clothes staying in your closet. But the universe tends towards disorder, so it will become more disorderly. And notice how the molecules of dye will actually end up at equilibrium, meaning equal amounts on both sides. Even though the molecules are still kind of going back and forth, they're not just going to stay there. They're still going back and forth, but they will tend towards equilibrium, meaning equal amounts on both sides. Now, how did the molecules move? Notice that they moved from an area of high concentration. High concentration means there is more dye compared to the water, to an area of low concentration. There was no dye on this side. And that's called diffusion. Diffusion, this is a really important concept. You're going to want to write this down. Diffusion is the movement of a particle from a high concentration to a low concentration. Remember, this right here on the left side is highly concentrated. There's a lot of dye compared to the amount of water. On this side, there's a low amount of dye compared to the amount of water, right? There's all water. Same thing here. There's a higher concentration on the left side of dye and a lower concentration of dye on the right side. So the molecules are going to move from a high concentration to a low concentration. But then here, notice that they're equally concentrated. And so now they're just kind of be moving back at the same rate. All right, I'm going to pause here and I'm going to show you a video about diffusion just so you can have it in your head, and then we'll continue on. All right, so again, diffusion is the movement of molecules from a high concentration to a low. So you notice here the concentration of the molecules were really high in the corner here, and then they diffused out to spaces where there is a less concentration of molecules. So think about if you were to spray perfume on one side of the room, there would be a high concentration of those perfume molecules on one side, and then they would diffuse out. They would spread out across where there's less dye, areas of less dye or lower concentration. And if we talk about it across a membrane, if we're talking about the movement of particles across a membrane, we call it passive transport. Let's think about why. Well, you're transporting. Things are moving across a membrane. So we see here on the left-hand side, we have these really tiny particles. And if you look closely, I believe those are water molecules. You can see the oxygen in blue and the two hydrogens, I believe. And notice how they are. It's kind of hard to place. They're moving across. But notice that the green molecules are a lot bigger. They're actually not able to get across the membrane. But basically, the water molecules are able to get across, so we call that passive transport because there's no energy that's needed to transport the molecules across the membrane. It's happening passively. There's no extra effort or extra energy involved. And that's going to be something that's really important for you to remember, this idea or this concept of passive transport. It's the movement of molecules passively across the membrane. Nothing's pushing it. It's just because of diffusion. Diffusion of molecules from a higher concentration to a lower concentration is basically the driving force behind the movement of these molecules across the membrane, called passive transport. Later, we're going to talk about something called active transport, which is the movement of larger molecules. So like, how do you get these green molecules across? You're going to need some help. You're going to need some energy. So we'll talk later about what that looks like. Now, let's say you have two solutes. So solutes are the particles that are dissolved in some solution. And so here we have orange and we have purple. And notice that over time, the purple molecules have a higher concentration on the right, but a low concentration on the left, so they're going to diffuse to the left. On the left-hand side, the orange has a high concentration here, but low concentration here, so they're going to go to the right. Now, it's important to note that the diffusion of a solute is not dependent on the presence of other solutes. So just because there are other purple molecules on the right side, because they're different types of solute, they're totally different molecules, they're not going to impact each other's diffusion rate, or they're basically like how much is diffusing on each side. So again, if notice here that the left-hand side more orange, and so the net diffusion is going to be to the right. Nutrition. And so eventually they will reach equilibrium, but it's not like these purple molecules are stopping the other ones from diffusing. In the same way, the net diffusion for the purple molecules is going to the left because it's going from a high concentration to a low concentration. The same thing here. Eventually they're going to reach equilibrium. So just like we said, each substance diffuses on its own concentration gradient, independent of the concentration gradients of other solutes. So a concentration gradient also, that idea is important. So a gradient, basically, think of a gradient as like, kind of like, have you ever seen people with like their hair is ombre, where they have like light to dark? So basically a gradient means like going from one extreme to the other extreme. So a gradient in this case would be going from high to low, right? It's moving down sort of, there's like a flow to it called a concentration. So the high flowing to low, kind of like your hair going from dark to light, right? That's a gradient. All right, I'm going to pause here and I'll continue back after nutrition. All right, so we've talked about diffusion, and we know that some molecules can easily diffuse across the membrane, the phospholipid membrane of the cell. But what about larger molecules or molecules that don't easily diffuse across the cell membrane? What happens to them? Well, we call this the semi-permeable membrane. The membrane is somewhat permeable. Permeable means things can pass through, things can get through. And the membrane allows a lot to pass through, but again, much of it needs to be controlled. So the membrane needs to be semi-permeable. Not everything is able to get in and out. So what are some things that need to get in and out? Sugars and amino acids need to get in to the cell. Those are the molecules that the cell is going to use for energy and to build new molecules. Lipids need to get in, and then I know it's kind of hard to see here, but water comes in and out of the cell, and then salts will be coming out and in, and then it flew away here, but we have a waste product of cellular respiration, ammonia, which also needs to leave the cell. If too much ammonia builds up in the cell, it can be really toxic for the cell. So stuff needs to get in and out. So let's think, here's our simplified phospholipid bilayer. And you can see outside the cell, let's say there are a bunch of lipids just hanging out outside the cell, and they wanted to get into the cell. Well, let's think about diffusion. Remember, we learned about diffusion is the movement of molecules from a high concentration to a low concentration. Let's think about this question. Which way will the lipids move? What will be their net movement overall? They're going to be moving back and forth, but overall, what's the net movement? Let's take a look. Notice here that they're going to end up moving from the outside of the cell into the cell. So if you go back here, there's a higher concentration of lipids on the outside, and they're going to be moving across their concentration gradient from high to low concentration. So they're going to be moving inside the cell. Now, some molecules can get through directly the phospholipid bilayer. So fats, lipids, they can get in and out of the phospholipid bilayer, or other nonpolar hydrophobic molecules. Take a moment and think of what nonpolar hydrophobic means. Nonpolar basically means there's not like two like charged ends of the molecule, or hydrophobic molecules can slip directly through. So these are things like oxygen or carbon dioxide or other fats. They can easily get in and out. But what about all the other stuff? How do they get through? So again, lipids can easily get in and out. But what about bigger molecules or hydrophilic molecules like sugar, amino acids, water? Remember, water is a polar molecule. It has a partially positive end and partially negative end. Ammonia and salt, these are charged molecules. And so how do they get in and out of this really hydrophobic membrane? Well, we know that the membrane is semi-permeable. Some things can actually get in and out. So again, some things are brought in, some things are brought out. And then also remember the cell is producing a product. They're producing lots of proteins inside through the endomembrane system. And so they need to excrete or export out products as well. So how do we do that? Well, the membrane has these holes or channels that allow hydrophilic materials in and out. So remember how we talked about hydrophobic molecules can more easily get in and out because the membrane, the inside here is hydrophobic. So hydrophilic molecules need a channel, something to get in and out. So we have, I know it's kind of hard to see here, but like water, this is a water molecule, or salts or glucose. These are hydrophilic molecules because they're polar, they have charged ends. And so there's these special channels that let them in and out. So we call this diffusion. So these channels, still diffusion is going to be happening. There's going to be molecules moving across their concentration gradient. So for example, here's our sugar molecules. Here's some polar molecules. And we're going to see that we start off outside the cell. There's a higher concentration of sugar. Inside the cell, there's a lower concentration. So think to yourself, overall, net movement, which way would the sugars move? So notice here, higher concentration to low. If you said from outside to inside, you're correct. Higher concentration to low. And they'll move through this channel. So they'll still diffuse. They'll move across their concentration gradient, but they're going to move through this kind of tunnel that's embedded within the membrane. So the cell does need to control what can get in and out. So it's not like all hydrophilic molecules or all larger molecules can get in and out of these channels. These channels are very specific. So each channel has a specific molecule that can diffuse in or out. So for example, there's a special sugar channel, there's a special amino acid channel, water channel, salt channel, ammonia channel that allows things to get in and out. So how do we build a membrane that's selectively permeable? Well, we started off, we already know that our membrane is built out of lipids, right? A phospholipid membrane. Don't give it away. But what molecule besides lipids, so think nucleic acids, carbohydrates, or proteins, can sit within the phospholipid bilayer or this hydrophobic bilayer? Think about a molecule that potentially could be hydrophobic. And again, that molecule is proteins. And I know we've already talked about this early in this video. But yes, the mystery molecule that is embedded, but we already talked about it, in the membrane are proteins. And why are proteins a good molecule for sitting, a good molecule for sitting within this hydrophobic lipid bilayer? Well, we know that's because proteins are mixed molecules. And let's explain. So remember, proteins are made up of amino acids, and different amino acids out of the 20, there are some that are hydrophobic, some that are hydrophilic based on the R group that's attached to the central alpha carbon. Go back to your macromolecules notes if you forgot about protein structure. But again, some amino acids are hydrophobic, meaning they don't like water. And so think, where would you find hydrophobic amino acids within this protein here? Here's the secondary and tertiary structure of the amino acid. We have these alpha helices, that secondary structure, bound together in tertiary structure. So notice that the hydrophobic amino acids are going to be the ones that are inside the membrane, so stuck in the lipid membrane. And what this does is it anchors the protein in. So it sticks in there because the hydrophobic interactions between the protein and the lipids around it will allow the protein to stick within the membrane. Then we can also have some hydrophilic amino acids sticking out on the outside, and they're stuck in the watery fluid in and outside the cell. Because remember, inside the cell there's water, outside the cell there's water, and hydrophilic amino acids, these are amino acids that are able to, they're not afraid of water. They like water because they have charged regions. And remember, water is polar, water is charged, so they can interact. And these outer regions here can be specialized as receptors for specific molecules. So depending on the chain of amino acids that make up these hydrophilic amino acid chains on the top and the bottom here that are sticking out of the cell, those specific molecules can receive other molecules. So basically they can bind to other molecules, and they're kind of like receptors that can now communicate information within the cell. We'll talk a lot more about that later this chapter. Okay, so again, here's another picture of our amino acids that are wound up in alpha helices. that then have our hydrophobic region within the lipid membrane and the hydrophilic within the cell and outside the cell in the watery spaces. Okay, so we talked about these proteins. They're called membrane proteins. Now, different cells, depending on what their function is in the body or for the organism, will have a different unique collection of proteins. So remember how we talked about how the proteins are specifically match different molecules. They can come in and out. Well, different cells have different requirements for like how much amino acids are coming in and out, or what other types of ions or molecules need to come in or out. And so again, if you compared the membranes of two different cells in your body, like membranes of your skin cells or membranes of like muscle tissue, you would find different assortment of proteins within the membranes. Let's see, we talked about this already, but we've got peripheral proteins on the kind of like loosely bound to the surface of the membrane. Integral proteins are actually in the lipid bilayer. And these are also called transmembrane proteins because they're across the membrane. All right, so we talked about passive transport. Remember how passive transport is basically the diffusion of molecules across the membrane to the other side, across a semi-permeable membrane. Now, this same thing we're about to talk about is also passive transport because there's actually no energy involved here. But we call this facilitated diffusion. Facilitated means there's something helping the diffusion happen. It's not just happening across the membrane on its own. There is something facilitating or allowing that to happen. Facilitate means like to help out. So if I'm facilitating you, I'm helping you do something. So these molecules here on the outside of the cell, notice that they're in a higher concentration outside. They're going to be diffusing to an area of lower concentration inside the cell. And these molecules typically are hydrophilic molecules or polar molecules that won't easily cross this hydrophobic membrane. And so this protein here will have, the inside will be hydrophilic or polar, allowing the polar molecules to easily get in. And no energy is needed, but these channels are made basically to make this hydrophilic space so that these molecules can get in. And again, they're moving from high to low concentration through this protein, which acts as a channel. These proteins, they're sometimes called globular proteins. They act as doors in the membrane. So they allow specific molecules to get through the cell membrane, like we talked about. And sometimes these proteins can actually close the door or open the door, depending on other factors, which we'll talk about. But basically, if the channel is wide open, if there's nothing blocking it, this can happen really, really quickly. So as long as you've got a higher concentration of the molecules on one side, they're going to easily, quickly diffuse. The same principles of diffusion, movement of solutes or molecules from a high concentration to a low concentration. But in this case, it's actually through some sort of channel. That's the only difference. So think about these proteins as kind of like the doorman. They're like the gates of the cell, allowing certain things in or out. All right, now water, remember water is a polar molecule. It's got its negative side with oxygen and the positively charged, slightly positive hydrogen end. And it wouldn't really be able to get through the lipid membrane, the really hydrophobic lipid membrane super easily. And so there are these special proteins called aquaporins that actually create this hydrophobic or hydrophilic region. So these amino acids have hydrophilic places here. And even notice these positive charges will kind of help pull in the water as it is diffusing in. And water can move super rapidly into the cell and out of the cell because of these aquaporins, these channels. So notice here's one part of the protein, here's another part, and so they're kind of acting as like doors, like two doors opening. And then here, these positive charge, notice the slightly negative side of the water molecule, its oxygen side, is attracted. It's facing towards the positive. So that protein is help pulling the water in. All right. Now, when we talk about diffusion, we're often talking about the movement of molecules or particles or ions through a semi-permeable membrane from high concentration to low concentration. Now, we're going to move on to a new topic, and this topic is really, really important. It's called osmosis. Now, osmosis is similar to diffusion, but it is simply discussing the diffusion of water. So remember, water is a molecule. So now think about the movement of water through membranes from high concentration to low concentration. Osmosis deals with water. Diffusion's dealing with other solutes or molecules or ions or particles that are dissolved in some sort of solution. So that's the key difference. But it's the same principle. And we'll take a look. So basically, osmosis, and you want to write this definition down. This is so important. Memorize it, know it, understand it. Osmosis is the diffusion of water, the movement of water, from a high concentration of water to a low concentration of water. So we're going to take a look at a picture that's going to show us what that looks like on the next slide. And this happens across a semi-permeable membrane. So remember, semi-permeable means some things can get through, some things can't, and a membrane is like a barrier blocking. And we use this equation called the water potential equation. We're going to come back to this, but we can talk about the water potential when we're talking about osmosis. Keep this little symbol in mind. All right, so let's walk through osmosis. So let's say we have this contraption here, and it's just kind of going to help us see the way that water is moving. And we have solutes. Solutes basically are molecules that are not water, that are dissolved in water. In this case, we're dealing with sugar. So sugar is our solute. And here we have on the left-hand side a low concentration of solute. Why is it low concentration? We'll notice if you count up all the sugar molecules, there's not that many compared to this side. There's a higher concentration of the solute on this side. Okay, so if you notice the sugar molecules, if you look really closely, they're too big. They can't actually get through the membrane because the membrane is semi-permeable or selectively permeable. It has selected that the sugar cannot get back and forth. But the cool thing is that the water is small enough that the water can get through the membrane. So how is water going to move? Well, remember, osmosis is the diffusion of water from a high concentration of water to a low concentration of water. So ignore the sugar molecules for now. Which side, or I guess they're relevant to this question, but we're not looking at the concentration of sugar. We're looking at the concentration of water. Which side, this side or this side, has a higher concentration of water? Basically, like which side has more water compared to solutes dissolved? Well, notice that this side on the left hand has a higher concentration of water. And so water is going to move from a high concentration across the membrane to a lower concentration. So you can see here, water is moving to a higher concentration from a higher concentration to a lower concentration of water. Now, it's tricky because when you think about concentrations, you think, well, no, this side has a higher concentration. No, this side has a higher concentration of solute, of sugar, but this side has a higher concentration of water. So if you were to look closely at all the water molecules, this side would actually have more water. Okay? So eventually, we are going to reach basically equilibrium. So at this point, now which side is more concentrated with solute? So now, basically, think about how much of these sugar molecules compared to the water on this side, compared to the concentration of sugar and water on this side. Well, we've actually kind of reached equal or similar concentration of solute. So basically, the water has basically like dissolved the solute or made the solute kind of like more spaced out. And you can think of it this way. The sugar molecule is a polar molecule. It has charged regions. And the water is kind of like going to attack. That's kind of how I think about it. That's not really what it's doing. But it is now going to attack, or it's going to latch onto those sugar molecules. And so when I think of osmosis, I always think of like water is going towards the direction of like attacking as many sugar or ions or solutes as possible. So again, if you look here, where are there more solutes? The water is going to go in the direction where there's more solutes. It's going to want to go attach on and dissolve and take apart as much solute as possible. I think of water as this like super, I don't know, like super excited to go and like attack and match up and bind with these polar molecules, these sugar molecules. So they're going to go find and attach onto them as best as they can. All right. So let's do some practice and then let's learn some more words to help us think about this. So let's look at these two solutions right here. So we have on this side, we have each of these yellow dots represents a molecule, one molecule of solute. And we have a semi-permeable membrane or selectively permeable membrane. And think, what can cross the membrane? Well, these molecules of solute, they're too big to cross the membrane. So let's say they're not able to cross. So let's say only water, the blue water can cross. What direction will the water move? What's the net movement of water? Take a moment, pause the video if you need. This is really good practice for you to think about. Look at your definition of osmosis. What is a definition of osmosis? Osmosis is the movement of water from a high concentration of water to a low concentration of water. Which side of this membrane has a higher concentration of water? Well, if you said this side, you're correct. There's more water on this side compared to the solute versus this side over here. So what is the net movement of water? Well, water is still going to be moving in and out across the membrane, but net is going to be towards the right. Basically, remember, the water is like going to go attack the other solutes. It wants to go latch on and dissolve and connect, because water is kind of like a magnet and it's going to go attach onto these polar molecules. So the water is going to be moving in that direction. So the direction of osmosis is determined by comparing total solute concentration. So remember, think about water's moves from a high concentration of water to a low concentration of water. Basically, water is going to move towards an area where there's a higher solute concentration. So some vocabulary, and this is something you want to write down. We're going to be using these words a lot, so it's going to be really, really important that you get these words in your head and memorize them, because when I say the word hypertonic, you're going to need to know what the word means. Okay, so a hypertonic solution is basically a solution like this right here, where you have more solute and less water. So solute, again, these are molecules, and there's less water compared to solute on this side. And we call this a low water potential. So water potential is low when there's less water. Okay, so write this symbol down, write this down, water potential symbol, less water, more solute, hypertonic. The way I think about this is hyper means like a lot, right? Hypertonic. So I think about solutes and I think about a lot of solutes are in a hypertonic solution. Hypotonic, these are solutions with less solute and more water. We call this having a high water potential. Write this down. So more water, less solute, hypotonic. And I think of hypo as just being smaller. I don't remember what the word hypo means. You can look it up. But I remember hyper means a lot of solute, hypo means less solute. So remember, the tricky piece is like thinking about if we're talking about solute versus water. And with these words, when we're saying hypertonic, we're talking about how much solute, a lot of solute, less water. Hypotonic, less solute, more water. So on this side, hypotonic, this side, hypertonic. And then isotonic would be a solution. We don't see it on this page, but if there's equal solute to water, that's just the water potential. So that's not high or low, it's just water potential. So iso means same, that root word. So write these down, memorize them. When I say these words, look at your notes and make sure you understand what they mean. Okay. All right, so let's do some practice. So let's say we take this cell, this sample cell. It's not really a cell, but it's kind of like our image that we're using. And let's put it in an environment. And let's add some sugar to the environment, or some carbohydrates. So we're going to take a look inside the cell. So on this side, we're going to add 0.03 molar of sucrose. That's a carbohydrate. It's a molecule. It's a particle, right? It's a charged hydrophilic molecule. It's a polar molecule. And 0.02 molar of glucose. So we've added some solutes inside our cell. So in total, we have 0.05 molar of solution inside the cell. And then let's look outside in the environment. So we have an environment of water, and we've dissolved 0.01 molar of sucrose, 0.01 molar of glucose, and 0.01 molar of fructose. So in all, we're measuring 0.03 molar on the outside. And this is just basically, when we're talking about molarity, we're talking about a measure of the amount of solute in a solution. And the solutes in this case are sucrose, glucose, and fructose. All right. So we're going to try to figure out what direction is the water going to be moving. Because remember, there's still water in here. We've got some water. We just dissolved sucrose and glucose in them on the inside. And then there's still water out here. We've just dissolved some other solutes in the water. So let's look at the cell. Compared to the beaker, the cell here, is the cell hypertonic or hypotonic? Remember what the hypertonic means. Hypertonic means a higher concentration of solute. Remember, solutes are these particles, sucrose and glucose. Basically, anything that's not water is a solute. Is a cell hypertonic or hypotonic? Take a moment, think in your head, Is there more solute in the cell or out of the cell? If you said hypertonic, you're correct, because the cell, if you look here, has a higher concentration of particles or solutes or sugars inside the cell. Now let's look at the beaker. Does the beaker a hypertonic solution or a hypotonic solution compared to the cell? Take a moment, think about it. If you said hypotonic, you're correct, because remember, hypotonic means a lower concentration of solute and a higher concentration of water compared to something else. So, which direction is the water going to flow? Is it going to flow from the inside of the cell out to the environment, or from the environment into the inside? Think about the definition of osmosis. Remember, osmosis is the movement of water from a higher concentration of water to a lower concentration of water. So where is there a higher concentration of water? Inside the cell or in the environment? If you said it's going to flow in to the cell, if you said that water will flow in, then you are correct. And why is that? Well, there's a higher concentration of water on the outside, right? There's less solute, there's less stuff dissolved. So if you were to zoom in really close and look at all the solutes, all the sugar molecules, and all the water molecules, there'd actually be more water on the outside compared to the inside. We're looking at the concentration of molecules. And the inside, we've got more molecules. We've got more sugar molecules compared to water. And so water is going to kind of move into the cell. Remember, it's going to go attack the other solutes that are present to try to go dissolve them. So water moves in. Okay, so how does this relate to cells? Well, cell survival depends on balancing water uptake and loss, because cells are often bathed in water. Like when you're thinking about your red blood cells, they're in this plasma that has a lot of water in it. And so depending on the concentration of water or other molecules or ions in your blood, that's going to change the direction of water flow. So let's look at an animal cell. Let's first look at the top here, and then we'll look at the bottom. And this is really good practice. This is something that you're going to want to make sure you understand for this unit. This is really important. So let's say you take a red blood cell. And remember, inside the blood cell, there's water. There's also ions and particles and solutes inside. And let's say you put it in a hypotonic solution. Take a moment. What does hypotonic mean? Remember, hypotonic means a higher concentration of water and a lower concentration of solute. So if there's a lower concentration of solute outside the cell compared to inside the cell, what direction is water going to move, right? Let's say there's more water outside the cell versus inside the cell. Remember, water moves from a high concentration of water to a low concentration of water. And so water will actually go into the cell, and the cell will become lysed. It will basically burst apart. It will expand so much like a water balloon that it will break apart. Isotonic solutions. So remember, isotonic solution means there's the same concentration of solute inside the cell versus outside the cell. So whatever, if you were to count up all the particles inside compared to outside, they'd be equal. So you have an equal amount of water coming in and out because it's at equilibrium. And then let's look at a hypertonic solution. So let's say, for example, you put your red blood cell in salt water. What would happen? Too much concentration of solute on the outside. Remember, hypertonic means that there is a higher concentration of solutes on the outside. So let's think, where is there more water? Well, remember, hypertonic means there's a lot of solutes outside. So that means there's more solutes outside and less solutes inside. So the inside actually is filled up with more water than the outside. Now, inside has more water, outside has less water. Which direction is water going to flow? Remember, water flows from a high concentration to a low concentration of water. That's osmosis, that's the definition of osmosis. And so water will go out and the cell will shrink and shrivel up. In the same way, let's look at plant cells. So plant cells, if you put a plant cell in fresh water, then what direction will water flow? Well, remember, we are putting in a hypotonic solution. So there's basically more water outside the cell than inside. Water moves from high to low concentration, so water will move into the cell. And when water moves into plant cells, it's actually stored in this spot in the center called the central vacuole. And the central vacuole will expand like a water balloon. And it gives something to the cell called turgor pressure, which basically means it makes the cell turgid. Turgid means it's really, really, really kind of like fully expanded out. So the cell walls, basically the central vacuole will push against the cell walls and it will be really, really, really tightly, I guess like the word is It'd just be the because the central vacuole is so big, it will keep the cell wall expanded out as far much as it can. Now, if you put the plant cell in isotonic solution, there's going to be an equal amount of water coming in and out, and so that central vacuole won't expand out as much, and we call that a flaccid cell. So basically, the cell won't be as like kind of expanded out. Like the central vacuole won't push against the cell wall as much, so it'll kind of be a little bit more wilted. And then if you put your your plant cell in salt water, then remember the salt water has a high concentration of solute on the outside compared to the inside. So there's more water on the inside compared to the outside. And water will flow from a high concentration of water in the inside out to go attack those solutes on the outside, remember? And so the central vacuole will shrivel up, and this is called plasmolyzed, which basically means the plant cell will shrivel, which is why you probably don't want to water your plants with salt water. Okay, so just to review, cells managing water. In an isotonic solution, remember that means there's the same amount of solute out and inside the cell. If we took an animal cell, here's an animal cell up here, and we immersed it in an isotonic solution, for example, blood cells in your blood, there will be no net movement of water because water will come in and out at equal rates. And the volume of the cell will be stable. So the same way for this plant cell, remember that central vacuole, that's the way I was trying to say it. The central vacuole controls the volume of the cell. So as it grows, as it gets more water, it will grow the volume and make it expand out larger. But if there's an even flow of water in and out, the volume will stay stable, and the cell will be flaccid, meaning it's not super turgid. It's not like volume is expanded out as much. Let's look at the hypotonic solution. So this is a review. So here's an animal cell, and remember, hypotonic means there's more water compared to solute on the outside. And so because there's more water on the outside, it will flow into the cell, and the cell will burst because it will expand out too much. Now, let's say some cells, like, for example, a paramecium, which is a bacteria cell, if you put it in pond water, then the paramecium is hypertonic. So there's a lot more solutes inside the paramecium compared to the water outside. And so because of that, water will go into the paramecium. Remember, there's a higher concentration of water. Let's say you put it in like this fresh pond water. Water will go into the place where there's less water, which is inside the paramecium. Inside the paramecium has a higher concentration of solute. And so because there's so much water coming in, these paramecium have evolved the ability to have this organelle called a contractile vacuole. And this contractile vacuole will actually pump water out of the cell. So even though water's coming in all the time, it'll pump water out using some energy, using ATP. So this costs the cell a lot of energy. And again, plant cells maintain water balance. Plant cells like being in hypotonic solutions. That's why you want to water your plants with like fresh water where there's not a lot of ions or molecules dissolved, because the inside of the cell will have more solute and less water compared to the outside will have more water and less solute. And so water will move from a high concentration to a low concentration and fill up the central vacuole and make the cell really turgid, or basically means like high volume. And then hypertonic, remember hypertonic means the amount of solute concentration is higher compared to water on the outside. So if you put a cell in a hypertonic solution, there'd be more water inside the cell than out, so water would flow out of the cell and it would shrivel up and probably die. Now, it's really cool because different organisms, like if you think about like different types of fish or aquatic organisms, they have evolved to be able to survive in their unique environment. So if you took a freshwater fish and you put it in salt water, it wouldn't live. And the reason is because of the way that their cells have evolved. And the basically like the tonicity. Tonicity means like what is the concentration of solute within the organs of the organism. I'll give you a picture on the next slide. But basically, animals have evolved the ability to kind of maintain their water balance based on the types of solutions that are in their organs. So saltwater organisms are hypotonic compared to their environment. If like a fish is floating in really salty water, then the inside is hypotonic, meaning the inside has more water compared to the outside because the water is so highly concentrated with salt or solutes. And so water is actually going to be flowing from inside the fish to outside the fish. And so they actually have to take up more extra water and then pump out extra salt because they're taking in more water. For plant cells, if you put it in a hypertonic solution, like you put it in really salty water, there would be more water inside the cell compared to outside, or a higher concentration of water. So water would flow out, and we have plasmolysis, which means wilting. Okay, so just like I was talking about a second ago, we call this osmoregulation, when different species of organisms are able to regulate their osmosis. So let's take a look at some examples. So let's look at a freshwater fish. So here on the top is a freshwater fish, and it's just floating around in freshwater. So there's a lot of just straight up water, not as much solute, not as much particles or molecules or ions. And inside the fish is a higher concentration of solutes and a lower concentration of water. So what direction is water going to flow? Well, the fish is drinking water, and it's also eating food, and so it's taking in ions. Ions are particles that are not water, so it's taking in ions, and it's also intaking some salt by the gills. So there is still some salt dissolved in the water. So they are going to be coming into the fish. Now, because the inside of the fish is hypertonic, basically what that means is there's more solutes dissolved in the fish compared to outside. There's more water on the outside than the inside. And so water will flow into the fish, right? Higher concentration of water on the outside will flow into the fish. So how does the fish survive and not just like blow up? Well, what it does is actually excrete. So it basically pees out a lot of water, and it's urine from its kidneys. So pees out a lot of water, and so it dumps out basically all the water that's coming in. So then let's look at saltwater fish. Saltwater fishes are in hypertonic environments, meaning there's more solutes dissolved. They're in salt water, right? And so the inside of the fish actually has a higher concentration of water than the outside. So remember, osmosis is the movement of water from a high concentration to a low concentration. So water will actually move out of the fish because it's moving towards a lower concentration of water. And at the same time, it's also gaining water and salt ions. Salt ions, again, are particles or solutes from drinking in the seawater. So what they do is they actually have to excrete. They have to get rid of a lot of salt because they're taking in a lot of salt water. They have to get rid of a lot of salt because they're taking in so much from osmosis. But they don't excrete a lot of water. So they don't pee a lot because they need to keep all the water they can get inside of them. Land animals, we live in a dry environment, and so we need to conserve water, right? Because even though we're not like swimming in water, there is water inside of us, and so there's a higher concentration of water in us compared to the outside, and so water easily leaves us, right? That's why partially why we sweat. There's water coming out of us when we breathe. We're breathing out water vapor, and so we need to conserve water in order to stay alive.